is Ladislav Vyklicky and come from Prague. And first of all, I would like to thank the organizers for inviting me. It was a great pleasure to come in here last night and last day I spent walking through Barcelona and really enjoyed the people. I walked 23 kilometers. It was a big walk. I really enjoyed it. Okay. I'm going to talk a little bit about molecular aspects of GRIN diseases. I will focus on patch clamp and molecular stuff. So some time ago, recent advances in high throughput DNA sequencing allow us to link the mutations and the disease, not only for GRIN diseases, but for many other diseases as well. And we somehow believe that by analyzing the consequences of the mutations for the function of the receptor, we can rectify the function and use some drugs that rectify the function in curing the patients. This is a simple link, but I will more or less focus on things which are pitfalls, which are not quite simple to overcome. So don't feel that I'm too depressed, but sometimes it will show that it's really not simple. I will give a brief introduction to mutations. I will talk about genes and GRIN genes, consequences for NMDA receptor function, surface expression, trafficking, and pharmacology. Ask the question whether we can replicate the pathology in humans, also in animals, in mouse, and I will talk a little bit about compensatory changes. All will be on a molecular level, and it will be no humans whatsoever, so maybe not fun for all of you. So if I go from the start, Adam and Eve were the first man and woman, and if the nature works really perfectly, then all the genes from Adam and Eve were transferred to offsprings, and we all be handsome and beautiful, intelligent, and we all would have a tendency to steal apples. This is not the case, because nature has introduced mutations, and each of us bears, when compared to our parents, about 50 to 90 de novo mutations. So we are not equal to our parents, but we have some additional information, and these mutations can affect perhaps any of 2,000 genes, which are important for our being. And these genes comprise also genes that encode for NMDA receptors. As mentioned earlier, there are GRIN1 and GRIN2 and GRIN3 receptors, channel types or subunit types, and mutation of these genes has been shown to be linked to intellectual disability, developmental delay, epilepsy, attention deficiency disorder, and many others. We may see these mutations as rare, and basically that only very few people are affected. This is, however, not the truth. In Europe, there is about 30 million people living with rare disease within the Europe. Rare disease affects 1 out of 2,000 people. It affects, the incidence is about 3.5% to 6% of the population. There are, in general, about 6,000 distinct rare diseases. Most of them start in the childhood, 70%, and most of them are of genetic origin. Of course, it has a big social impact. 7 out of 10 patients and carriers have reduced or stopped their professional activity. 8 out of 10 patients have difficulties completing daily tasks. As I mentioned, I will talk about molecular stuff. So what we do in many cases is we take a gene, either human or rat, which are quite similar, mutate it and trying to investigate what's going on with the receptor function. So this is the vector. We transfer human embryonic kidney cells that don't express NMDA receptors, and we then investigate the properties of these receptors. In some cases, we use also native receptors from hippocampus of mice and culture them in an islet culture system, which is one neuron per a dot. In the patch-cam technique, we use the system. We have 10 tubes like this filled with different solutions. We apply quickly different solutions filled with different agonists or compounds and investigate the properties of the responses. So like in here, so we can do the dose response curve and see what happens with the dose response curve for glutamate, or we can do the pharmacology. I will take an example from our recent study that hasn't been published yet, where we studied de novo mutations in the C-terminus of the receptor. C-terminus is not essential for the function itself. If you delete C-terminus, the receptor still works somehow, not properly, but works. C-terminus has a size where it's important for trafficking the receptor to synapse, has palmitylation, site phosphorylation, and other sites. And we selected about, I don't know, 10, 15 de novo nonsense mutations, means that the mutation introduces stop codon, and from that side, further the protein is not translated. And these mutations are linked in here, and some of them are associated with autism, autism spectrum disorder, epilepsy. These are the green ones, intellectual disability, red ones, and abnormality of the central nervous system. So already from this first slide, you can see that the spread of the disease is not equal to each subunit, but we look what's going on with the function of the receptor. So it's just briefly shown in here. For many mutations, we haven't seen much change, or no, not obvious change. But for some mutations, particularly those that are close to the membrane domain of the receptor, there are changes in the function of receptor, particularly increase in the affinity for glutamate and for glycine, increase of desensitization, and changes in the probability of opening. We don't need to remember all of these are not really important, but just if you have a look on this, can you imagine drug that would fulfill all these four criteria? Increase the glutamate affinity and reduce the affinity for glutamate and glycine, rectify the desensitization, and increase the probability of opening. This is not a simple approach to fulfill all these criteria. In the next mutation, which is quite close, we have only change in the in glycine, an increase in the glycine. But if we go to subunit 2A, we have increase in the sensitization and change in probability. So for perhaps each of the mutations, we would need a slightly different drug or drug combination. Maybe probability of opening is the ability, if you activate the channel or receptor is supersaturating concentration, the channel is not always fully open. So the relative percentage of the channel is being open is the probability of opening, and is different for each subunit combination. For combination which comprises of 2B subunits is about 10%, for 2A it's about 30%. Maybe you may argue that this is a special case for receptors or mutations or that are in the C-terminus, that is not in general. So we took a lot of papers, many, maybe 50, and derived took the affinity, which is published, and how it correlates with the glycine and glutamate affinity. So in many cases, it's the glycine affinity is the only function that is changed. In many cases, only glutamate. But in many cases, like in here, both glutamate and glycine affinities are changed, or either increased or decreased. In exceptional few cases, this is disproportional. One is increased and the other one is decreased. Okay. While we couldn't find much or relatively small changes when we did the patch-chem studies. So now let's have a look on other aspect of the receptor. Receptor has to be born somewhere in the cell and be transported to the cell membrane. And this transport is complicated, has many steps, and not all the receptors are transported to the membrane. And we can measure relative distribution of the receptors to the membrane. How is this in wild type, like it's always one in here, and how is it in mutated receptors? So you can see that for the 2A subunit, for the one that is almost at the end, if we delete it short trunk, it's increase the surface expression, but for most of them, the surface expression is significantly reduced by 70%, a lot. In the case of 2B subunits, the reduction is considerably less, but for all of them, the concentration or density of receptors in the membrane is reduced. We perhaps all believe that not the surface expression, but the receptors that are in a synapse are important. So let's have a look on expression of receptors, how they get into the membrane, into the synapse. So we did specific staining of receptors in the synapse and look how the density of receptors in the synapse is affected by de novo mutations. And you can see that for most of them, the density is reduced. So if we have a complicated situation when we have a reduced number of receptors in the membrane, reduced number in the synapse, and increased affinity for glutamate and glycine. So and we don't have simple treatment which will allow increased transport of the receptors to the membrane or to the synapse. In the next step, let's look what happens with the pharmacology of receptors. In my lab, we studied the effect of steroids or neurosteroids. Neurosteroids are steroidal compounds which are normally produced in the brain and act locally in high concentrations, and they affect all type of receptors, including receptors for NMDA, and they have a non-genomic action. And the first who described the non-genomic action was Hans Bruno Seily, who put allopregnanolone to rats. They immediately fell asleep, and after a few minutes they wake up and behave normally. So there are three classes of steroids. Ones that are inducing potentiation is pregnenolone sulfate. These are flat. Bandit, that are pregnenolone sulfate, that are inhibitory, and cholesterol, which is essential for the receptor function. If you deplete the cells or neurons of cholesterol, and NMDA receptor usually don't open at all. So let's have for the nonsense mutations, let's study and look whether we would be able to rectify their function by the steroids. At least if we use pregnenolone sulfate and pregnenolone butyric acid, so which is more potent synthetic compound. So you see that we can activate the receptor more, boost their activity, and that it works for many mutations, but not for all of them. Those mutations that are close to the membrane, suddenly the potentiating effect of steroids is changed to inhibitory, and for butyrate, the potentiating effect is almost lost. It with more complex situation occurs in the case of 2B subunits, where there is a systematic change of each subunit. The potentiation is slightly different. While for wild type is in here, for the truncation, it increases from 200 potentiation almost to 400, and then it drops to almost zero. So one has to basically, if one decides to use steroids, one has to very carefully analyze what the steroid will do with the receptor and how pronounced effect there'll be. Again, you may argue that this is the case of C-terminal domain, that this may not be general. I took another example from our different study where we studied mutations in the membrane domain, and here are the results. These are the mutations in membrane domain. This is membrane domain of the receptor, and here are the mutations. You don't need to know which one is which, but you just see that the relative potentiation induced by pregnenolone sulfate and butyrate are quite different. In some mutations, the butyrate doesn't work almost at all. For some cases, the potentiation effect of pregnenolone sulfate is changes to inhibition, and for some cases, the potentiation effect is considerably stronger than for the wild type, almost doubled. Okay, so what we somehow miss is this is a molecular stuff which is quite exciting to do, being biophysicist, but for the biology, one would like to have a more biological model when we could try to use to see more complex behavior. So what we did is we generated GMO mice, which has a mutation within leucine to valine at position 825. It is in the membrane domain of the receptor. This mutation has been described for a patient who was a boy suffering for intellectual disability and autism. It was the boy started to walk late or later, never spoke, never started eating on his own, and never played with his toys. And so we generated these mice, and we were eager to see whether we can replicate the same symptoms as in the humans. The mutation in the membrane domain causes actually reduces probability of opening. When it's homozygous, it drops the probability from 10 to almost 1. When heterozygous, which is the case of humans and mice, which we studied, it drops about by 50% to about 5%. And it is isolated change. No change in surface expression, no change in delivery of the receptor to the membrane. We looked on the molecular aspects, and the neurons from heterozygous mice, they expressed a little bit less of NMDA receptors. The currents were usually smaller. In contrast to ampakainate, which were exactly the same, the amplitude of postsynaptic currents was exactly the same in mutated and wild-type mice. Only the rate of deactivation was shorter in the mutant mice. This is a typical feature for 2A subunits, which has faster deactivation. Okay, now we looked on the behavior stuff, which I was eager to see some form of behavioral changes. So it was done here, are shown 10 different tasks which the mice was treated with, and basically, with exception of prepulse inhibition, there were no significant changes. These were significant, but the significance was only marginal, no huge changes. It was confined only to males, not big change. The prepulse inhibition was perhaps the only change which we could see at this stage. The mice behave normally, they breed, they play, they build nests, completely normal. So it was a big frustration for me not to be able to replicate at least some of the changes which I was expecting to see. So then I was offered an additional test. It was intelligent case, which is a test where the mice can freely move, as in natural conditions. and has to find a corner with the water, and remember which corner is it. And again, this test was exactly the same. The ability to learn was exactly the same from the wild type and for the mutant mice. And then the last chance was to use what's called reverser, to change the position from the position where it was learned to a new position. And in this test, the mice failed. They were not able to learn the new position with difficulties, and it was difficult for them to learn the new position. We don't know why this is so. Maybe there are some compensatory changes. Maybe it may be interesting to see that, because this is in general that patients which have the same mutation, they don't usually have the same symptoms. So sometimes it's there may be a number of reasons for this. Compensatory genes, compensatory changes. Maybe we are not looking on proper things which are where NMDA receptors are involved. We are usually look for synaptic transmission, properties of the receptors, like for excitation, but NMDA receptors are involved in proliferation of the cells, migration, neurogenesis, growth of axons, synapse formation, pruning, metabolic role, and so on. So we don't know, and the basic research data were not able to analyze all these features yet. Okay, one possibility is that there are also some form of compensatory changes, that this is not only the receptor which has impaired function, but maybe there are changes in gene expression of surrounding or neighboring genes. And for this purpose, we use zebrafish, which has also NMDA receptors with similar structure to humans. They are highly homologous, and we deleted to A genes and look for the phenotype. And indeed, these fish swim considerably less at the age of 6 days. And this is a duration of 1 hour. And you see the difference between the mutated receptors and wild type receptors, quite obvious change. And we looked on the qRT-PCR expression of genes encoding for NMDA receptors. And what was amazing and was quite interesting to see that in mutants, the receptors, basically all subunits, are expressed much more, like GRIN1 subunit encoding for GLUN1 subunit is expressed twice as much. Gene encoding for 2A, three-fold more. B subunit, again, many more times. So maybe in this type of deletions, the problem is not lack of function, as one would expect, but rather gain of function, that more receptors are being expressed. And indeed, usually these deletions are associated with epilepsy, which is something what increases the function of the system. In conclusion, I would like to say that our results and results of many other people show that mutations usually alter the multiple functions of the receptors. Usually, there are about 6,000 distinct rare diseases, but there are only 400 registered orphan drugs. So we desperately need more drugs that could be possibly tested and used in clinics and investigated in basic science. Maybe drug repurposing may be the way. Using steroids as a tool in pharmacological studies indicate that their modulatory effect is substantially different for individual disease-associated variants. Therefore, personalized medicine is required. And finally, that compensatory protein surface expression and gene expression changes should be considered when deciding whether the given variant is a loss or gain of function. I would like to acknowledge my co-workers. Some of them were from Spain. Miriam did most of the work on rats or mice, together with Teresa and Clavinda. Victor did the surface staining. We have people that did molecular stuff, which Agnieszka did the behavior stuff. These people, Eva, Anka, and Barbara, did the synthesized steroids for us. Aleš did the molecular stuff. And this is perhaps all, and I thank you for your attention.